The cerebellum is located at the base of the brain, dorsal to the medulla oblongata at the height of the pons. It is divided into 3 lobes. The anterior lobe, which is separated from the posterior lobe by the primary fissure, and the posterior lobe, which is separated from the flocculonodular lobe by the posterior lateral fissure. Despite contributing only to about 10% of the volume of the brain, it contains about 50% of the total number of neurons in the brain. Similar to the cerebrum, the cerebellum can be divided into an outer region of gray matter and a central area of white matter. The gray matter corresponds to the cortex of the cerebellum, which contain many of the body and the dendrite of the cerebellar neurons and can be divided into 3 layers. The outermost is the molecular layer, followed towards the interior by the Purkinje layer and then the granular layer. Then the region of white matter that contains myelinated axons and neurons. Towards the center of the white matter we find the deep cerebellar nuclei, which are the only structures from which the outputs of the cerebellum originates. Each nucleus receives input from a given area of the cerebellar cortex. All inputs of the cerebellum comes either from the medulla oblongata or from the inferior olive. The inferior olive is located outside the cerebellum, below the pons, within the medulla oblongata. An important role of the cerebellum is to ensure that the execution of each movement is done correctly. To ensure that this happens, the cerebellum compares the signal sent from the motor cortex to the limbs with a signal that reports back from the limbs regarding the position of the limb as the movement takes place. If discrepancies are identified, the cerebellum generates the information necessary to correct the movement. To better explain the structure of the different layers of the cerebellum, we're going to expand the view of the cortex and the white matter. Here we can observe the multitude of cells making up the cerebellum, but at this density it would be impossible to appreciate the working relationship of each cell involved. Therefore, we're going to see a very simplified version of a single functional unit out of the approximately 16 million existing in a human cerebellum. Each individual functional unit is made up of one Purkinje cell as the central controller, which is fed information by climbing fibers, thousands of granular cells, and regulated by many interneurons. As mentioned before, the outermost layer of the cerebellum is the molecular layer, which contains the dendrites of the Purkinje cells, as well as the axons of the granular cells that are organized in parallel to each other, forming the parallel fibers. The parallel fibers run perpendicular to the dendrites of the Purkinje cells with which they synapse. Parallel fibers also synapse with dendrites of the interneuron stellates and basket cells, whose bodies are located in the molecular layer. Finally, the tips of the dendrite of the Golgi cells are also located in the molecular layer. Under the molecular layer is the Purkinje layer, which contains the body of all Purkinje cells as well as the body of housekeeping glial cells, and part of the dendrites of the Golgi cells, and part of the axons of the granular cells. Below, under the Purkinje layer, we find the granular layer, which contains the body of billions of granular cells which project their axons into the molecular layer. Here are also located the body and axons of the Golgi cells. Below the cortex is the white matter, which is made up of all the axons of the climbing fibers and the muscle fibers which bring information to the cortex. Lower, within the white matter, we find the deep cerebellar nuclei, which receive information from the Purkinje cells and from the inferior olives and send signals to the frontal motor region of the brain. A multitude of cells organized in a much prescribed fashion operates in a specific sequence to carry out the comparison between the initial instruction for a given movement with the actual movement and then generates the required correction. The main input to the cerebellum is through climbing fibers which come from the inferior olives and synapse with the dendrites of Purkinje cells in the molecular layer of the cerebellum. The other input of the cerebellum comes from mossy fibers coming from the medulla and spinal cord and synapse with dendrite of granular cells. Synapse which is modulated by Golgi cell in a unit called cerebellar glomeruli. A very simplified sequence of electric signals required to achieve the above-mentioned correction of the movement is as follows. We're going to use a limb as an example. The motor area of the cerebral cortex sends a signal to the muscle of a limb to execute a given movement. This signal is copied to the inferior olive, which then, using climbing fibers, convey the same information to the Purkinje cells. The inferior olives also send a copy of the original information to a corresponding deep cerebellar nucleus. At the same time, the proprioceptors of the muscles in the limb in charge of carrying out the movement send feedback signals reporting the position and rate of movement of the limb to the inferior olive. This information is then transmitted to the deep cerebellar nucleus and to a granular cell. This granular cell then carries the muscle signal through their axons, which form the parallel fibers in the molecular layer of the cerebellum, to the dendrites of the Purkinje cells and to the interneuron stellate basket as well as Golgi cells. Further complementary information from the vestibular system and from the spinal cord are also carried through mossy fibers to the granular cells, so it reaches the Purkinje cell. Up to now, all of the above-mentioned connections are stimulatory signals. The information gathered in the Purkinje cell is then modulated directly by inhibitory signals generated by the interneuron stellate and basket cells located in the molecular layer and indirectly by the Golgi cells through the connection in the cerebellar glomeruli mentioned before. From all of these synapses, which can be as many as 200,000 for each Purkinje cell, a single final output in the form of an inhibitory signal is generated by the Purkinje cell. This signal is then sent to the deep cerebellar nucleus, which, using for comparison the initial information from the olives as well as the information from the muscle, generates a new correcting signal, which is then sent to the premotor area of the cerebral cortex via the thalamus. Based on this information, the cortex sends a new modified signal to the muscle in the limb to correct the ongoing movement. This sequence is repeated multiple times until the movement is completed.